Alright, welcome back. So we're moving on to our next topic, which is nucleic acids and proteins. So again, we have our key knowledge checklist that comes from VCAA. Strongly recommend that you print this slide here off and tick these things off as you feel comfortable with them. But what we're going to be covering is nucleic acids and the instructions that they contain to make proteins for cells, different types of proteins. So we're going to be looking at the proteome, which is the different types of proteins that we have, the levels within these protein structures, so primary, secondary, tertiary structures and those types of things. We're going to be looking at making a polypeptide chain from amino acid monomers, the structure of DNA, its subunits, all those types of things, and the genetic code, so going from DNA to RNA before we're able to make that protein. So starting off with nucleic acids, they are the long molecule that's made up of 3 distinct chemical parts, and they are what store the information to be making our proteins that we're going to be looking at a little bit later on. These nucleic acids are the genetic material that make up every single living organism, and what you're going to have is either DNA, deoxyribonucleic acid, or RNA, ribonucleic acid. So DNA and RNA are both versions of nucleic acids. They are genetic material and they contain the codes to make proteins. So what we do is we go from DNA, which is made up of our phosphate sugar bases, which we have groups of here, remembering how they pair together. And these bases combine together to make our DNA. Sections of our DNA make up genes, and these genes contain information to make a specific protein outcome. And a strand of DNA together, when it condenses, is what we call a chromosome. So when we looked at, say, genetic diseases in year 11, we were looking at chromosomes, chromosomal abnormalities. So looking at Down syndrome, having extra chromosomes, having less chromosomes, all of those types of things. So DNA is the smaller part that makes up chromosomes as a whole, and genes are sections of DNA. So here we go again with these nucleotides. They are the monomers that bind together to make the nucleic acid polymers. So nucleotides combine together to make DNA or RNA. They have the 3 chemical parts that we spoke about before. So we're looking at having the 5-carbon sugar, depending on if it is DNA or RNA. We have a phosphate group. And then we have the base depending on if we are looking at DNA or RNA. So we have A, G, C, and T. And remember that in RNA we have U instead of T. It's important to be able to switch quite readily between DNA and RNA. It comes up very frequently in exam questions where they will give you DNA and ask for the RNA, or they'll give you RNA and ask for the template strand of DNA, those types of things. So you need to know how these things bind together. So A generally pairs with T, except for in RNA, where A will bind with U. So nucleic acids are sets of instructions that we spoke about before. So my DNA codes to make the proteins. And every cell will make different proteins depending on what its function is. So some proteins that will be made will be very similar in cells, others will make very different cells. So in order to get from DNA to my protein, my DNA needs to be read. And it gets read in terms of 3 bases grouped together. So each set of 3 bases is what we call a codon, and that codes for 1 amino acid. So the proteins are different depending on the arrangement and the number of those amino acids. So we have 20 different amino acids, and that's allowed for with the coding of those 4 different bases. So we have 20 different amino acids. The coding is very important. We start at different places and stop at different places on our DNA strands depending on which protein we are going to be making. The order of those amino acids can change. And again, this is where we spoke about some of those mutations in year 11, where if we don't have So say we change one base, we have a point mutation, then we end up with a different amino acid. If we have a different amino acid, we have a different protein chain, which means it might be a completely different protein. It might not be able to function the same, or the one base change might not change it at all, and you still end up with the same product. So we go from DNA to RNA, and this is a very important part of our process in terms of making the protein. So ribosomes can't read DNA to make the ultimate protein that we are looking for. So in terms of reading those triplet codons to attach the amino acids, it needs to come from RNA first. So the first step is to convert my DNA to RNA, and my important enzyme in this one is RNA polymerase. We have a template strand and a coding strand of my DNA, and my DNA gets unwound from each other. So it's like separating the two sides to a ladder, but keeping the ends intact, so maybe like sawing it in the middle, so that we can then add on these nucleotides to build the RNA strand, and then it gets rewound as it goes through and past. So transcription, there are 4 steps to transcription, and it's quite important that you understand these. The first one is initiation, and transcription is the first part of this process. It is when we are going from DNA to RNA. Okay, DNA, RNA, that's transcription. Initiation, RNA polymerase binds to the promoter start on the template strand. So basically, initiation, RNA polymerase is going to bind to my DNA. I then get elongation. The RNA polymerase moves along that template strand, and we're making that new RNA molecule that we were just talking about. So we're looking at adding in uracil where we would have had thymine before, and making that new RNA strand. Termination, a loop forms at the end of the RNA molecule, and that detaches itself from the copying strand that it was on the DNA, so that the DNA is then able to reconnect with each other, and there's very little disruption or damage caused to the DNA that we started with. And then we have processing as the 4th step to transcription, where introns are removed. And so introns are unimportant bits of DNA. They're not needed to make the final product. They're kind of think of them as like filler pages. So even if you're reading a textbook or a story or anything at all, not every single word, even these PowerPoint slides, not every single word in this PowerPoint is vitally important. The introns are those bits that are kind of filler, stuff that helps everything else make sense, but it's not really that valuable. What we're looking for is these exons. They contain the key essential information. They are the ones that are coding for the actual protein outcome. So we take out the useless introns, we're left with the exons, and we splice them together. So we stick them back together, and that's where we get mature mRNA. So the mRNA that we have up here in elongation is not mature mRNA. It's pre-mRNA, so before these changes happen to it. And it's a molecule that consists of the single protein coding sequence for the protein outcome that we desire. We then add a methyl cap and a poly A tail to help finalize our mature mRNA message, and that's transcription. So in terms of key terms that you're going to need to make sure that you really know, we're looking at template strand, the strand of DNA that the RNA copy comes from, introns, the non-coding regions of DNA, and exons, the function region of DNA. So they're the important bits. And as you can see here, we've got our pre-mRNA, we've got our intron bit in the middle, and we just take that bit there out, stick these 2 red bits back together, and we end up with our mRNA at the end. So now we have our mRNA. We're going to be looking at making our protein. So this is where we look at our triplet codons that we have here and matching them up with the amino acids. So there are lots of different steps to this stage. This process is what we call translation. And in this one, mRNA leaves the nucleus. So all of transcription occurs inside the nucleus. Translation occurs outside of the nucleus. And it's where we're translating the message. Think like translating between languages. You completely change it. We're translating it from a nucleotide language to a protein language. That's what we're translating here in this process. So mRNA leaves the nucleus, step number 1. Step number 2, the ribosome binds to mRNA at a specific area. So you think back to when we looked at cells and cell organelles and things like that in year 11. You've got free-floating ribosomes, and you've got ribosomes on your rough ER, your rough endoplasmic reticulum. So your mRNA can bind to those ribosomes in both of those locations. So the ribosomes then read your mRNA strand, so they move along it, and they start to match tRNA or transfer RNA anticodons. So they've got a sequence of 3 bases on them, and those 3 bases match up to the 3 bases on DNA. Sorry, not DNA, your RNA. So the 3 on tRNA match the 3 on the mRNA, and the tRNA carries in the amino acid. So each time a new tRNA comes in to read the next triplet, so to match up with the next triplet on the mRNA, the 2 amino acids, the one from the one that was there previously and the new one that's just been brought in, combine together, so they stick together, and we end up making this polypeptide chain from those amino acids. So as the amino acids bind, the tRNA disconnects from the mRNA and it goes to regain its amino acid so that this process can continue. So the ribosome will keep reading until it hits a stop sequence in the mRNA, and then it releases the polypeptide and the mRNA. So remember, my polypeptide is that sequence of those amino acids. It's my polypeptide chain. My mRNA can just keep going about its job, so we might be making multiple proteins from this one, or the message might be recycled, so it might be broken down to be remade into something else. The polypeptide forms into its native shape, and we're going to be talking about protein structures shortly, so we're looking at the different stages within that, for it then to become a functional protein within the cell. So just after translation, it's not yet functional. There are other things that need to happen to it before it can do its job. So this is what we have here. We have our mRNA at the top. We then have our ribosomes, and these guys in here, these 3 are tRNA. And you can see that each one of them has a codon from the mRNA and an anticodon that's on the tRNA. And they match together. The tRNA is bringing in the amino acid at the end here. The next one across comes in, and these 2 amino acids are going to form a polypeptide bond. So here, these guys here are going to join. The next one will come in, and we'll be adding them on and on and on again and again and again until we get this polypeptide chain of all those amino acids together. So this is a diagram that is very common to your exams, and this is how we work out the amino acids that correspond to the codons that we were looking at on our mRNA. So when we're looking at our mRNA, we read it in terms of the first nucleotide, so the first base that we have in our triplet codon. Then we read the second base, and then we read the third base. And as you can see here, there's the stop codons. These are where the RNA polymerase will stop reading, and then it will detach, so that then you get your completed amino acid chain, that polypeptide chain. So to read them, you read the first one here, which takes you to this row. So say you had a C for the first one, an A for the next one, and a G for the last one. So you would go across C, then your next one is A, so you would read all the way across. So you'd CA, and we wanted CAG, so we'd make a GLN amino acid. There are several different ways that you can see this. You'll see it as a table. You might also see it as kind of like a pie chart where you read from the outside towards the middle and it tells you your amino acids. We read from the middle towards the outside. So you start from the middle and you work your way out and it tells you what your amino acid is. They are very they are very common in your exams, and I recommend that you practice reading them and converting your mRNA to its matching amino acid to make your polypeptide chains. As we stick the amino acids together, we form something called a peptide bond. Just between these guys right here, that's the peptide bond. And what happens as a result of these peptide bonds is that we produce water. So anyone that does chemistry and looks at these reactions quite heavily, you should get this quite quickly. It's a condensation polymerization reaction. So condensation is because we are producing water, and polymerization is because we are making a polymer. We are making a polypeptide chain. So if we have a look at the next one, as you can see here, we've got two amino acids. They join together. We add another amino acid. It joins to it. And this group here should look familiar. H2O. Another amino acid, another group of water. And this process just continues and continues until we finish looking at our protein chain that we're looking at making, and we produce water. It's our polypeptide production condensation reaction. So we've looked at going from DNA, and DNA does that replication process. We did look at this last year in terms of producing new cells. And then in terms of making a protein and it's doing its job, we first go through that process of transcription, so converting DNA to RNA, and then translation, RNA to a protein. So we have three different types of RNA, and it's important that you recognize the differences between these three. They were mentioned in the study design. So you have messenger RNA, transfer RNA, ribosomal RNA. So messenger RNA is our mRNA. It didn't work very well. mRNA. Transfer RNA is our tRNA, and ribosomal RNA is rRNA. And ribosomal RNA is just RNA that makes up a ribosome. So it helps to convert the mRNA into that amino acid chain, and it's where proteins are synthesized or made. So the proteome is the entire set of proteins that can be expressed by a cell, tissue, or organism. And we're looking at different types of proteins here. So we've got fibrous and globular proteins. They're the two main groups. And within them, we have different types of protein again. So collagen is an example of a fibrous protein. Our enzymes that we use are globular proteins. Antibodies and protein channels and hormones are again all globular proteins. So protein structure, we have 4 different levels of our structure, and you do need to know the differences between them. This was the very first short answer question in last year's VCAA exam. They had to identify the different primary and secondary structures that were visible in a protein structure that they were given. So the primary structure is the sequence of the amino acids. So it is just that polypeptide chain. Nice and easy. We've spoken about this one earlier on in this PowerPoint. Primary structure, polypeptide chain, the sequence of the amino acids. The secondary structure is where we start to get that folding happening with those amino acids. So there are 3 different structures to secondary structure. We have alpha helixes, which are these guys here. They're like coils. We have beta pleated sheets, which is this guy here. Kind of goes up and down. And then you have random coils. Sometimes it's referred to as a random walk, where it's kind of a little bit of everything and not quite really that much of a structure. So if it doesn't sit with an alpha helix or a beta pleated sheet, it's going to be a random coil or a random walk. The next level is the tertiary structure, and this is where we start to get some more bonds happening. So we're looking at disulfide bridges where sulfates stick together. We can look at having hydrogen bonds, ionic bonds, all these different types of bonding that's happening across those amino acid chains. So every amino acid has a slightly different structure, and the bits that stick out from them is what causes these types of bonds to occur. So it's the interaction of the secondary structures, really. So it holds its place by the bonds that we mentioned. Amino acids that were distant in the primary structure can now become very, very close to each other. So something that could have been quite a fair distance away in terms of it being a straight chain are now very close together because they curl around and become close again. So the subunit for a more complex protein has now been formed, and this is where we kind of work out whether it's going to be fibrous or globular, because it now has a more functional shape. It's now able to do a job. Quaternary structures is the next level again, and this is the packaging of those proteins to form the final protein complex. So in the tertiary one, you kind of get a final product, but sometimes you need multiple proteins together to do a job. And we're looking at making glycoproteins, so adding carbohydrates into proteins. We look at combining multiple protein subunits together. So this one that we have here is hemoglobin, which is 4 proteins together. So, and then we've got 2 alpha and 2 beta polypeptide chains to make up one hemoglobin protein. So it's 4 to make 1. So quaternary structures are multiple tertiary structures together, and it's when you get carbohydrate and other groups added into your protein.